Good morning, it's October 3rd, and this is your daily brief in science. Here's everything you need to know. Neuroscientists have achieved a significant milestone by creating the first complete wiring map of the fruit fly brain. This comprehensive map details nearly 140,000 neurons and over 54.5 million synapses, showcasing the intricate connections between nerve cells. The research, co-led by Malamurthy and Sebastian Seung from Princeton, was published in a special issue of Nature on October 2, 2024. It involved collaboration among researchers from Canada, Germany, and the UK under the project known as the Flywire Consortium, which included over 287 researchers and volunteers dedicating an estimated 33 person-years to ensure data accuracy. Advancements in AI computing played a crucial role in reconstructing this wiring diagram, transforming what would have been an impractical task into a feasible one. The team analyzed more than 7,000 electron microscopy images of thin brain slices and employed machine learning to assist in aligning neuron structures with human oversight for error correction. Sebastian Seung remarked that initial skepticism about the feasibility of creating a connectomy has been overcome, revealing valuable insights into the cellular details of brain function. The study identified distinct circuits for various functions, such as movement and vision, exploring how these circuits interconnect to enhance our understanding of brain functionality. Dr. Gregory Jefferies emphasized the importance of understanding how brain cell networks facilitate interactions with the environment, underscoring the significance of this research. The open-access nature of the project promotes broad scientific collaboration and encourages further exploration of neural mapping. Despite extensive research, knowledge about AVP neurons has been limited due to the absence of a complete circuit diagram, which this study aims to address. The mapping process has been likened to creating a Google Maps for brains, providing detailed annotations that aid in identifying over 8,000 cell types. Astronomers have confirmed the discovery of an exoplanet named Barnard B, located just 6 light-years from Earth around Barnard's star, the closest known star to our solar system after Alpha Centauri. This discovery marks a significant milestone in the ongoing search for Earth-like exoplanets, particularly because Barnard's star, a red dwarf, is a prime target due to its proximity and habitable zone being closer than that of hotter stars. The confirmation of Barnard B required 5 years of observational data, highlighting the challenges astronomers face when detecting distant planets. Utilizing the European Southern Observatory's ESPR-ESSO instrument, researchers measured the wobble of Barnard's star caused by the gravitational pull of orbiting planets. Despite this breakthrough, Barnard B is inhospitable for liquid water and life as we know it, with a surface temperature of around 125 degrees Celsius due to its close proximity to its star. In addition to Barnard B, evidence of 3 other potential exoplanets around the same star has been identified, although further observations are necessary for confirmation. The search for planets around Barnard's star has been ongoing since the 1960s, with previous claims often disputed due to interference from stellar activity. The discovery of Barnard B is significant as it represents progress in identifying small exoplanets, which are notoriously difficult to detect. Barnard B is one of the least massive exoplanets known, with a mass between 0.17 and 0.32 times that of Earth, and it completes its orbit in just 3.15 Earth days. The findings were published in the journal Astronomy and Astrophysics, reflecting a growing interest in low-mass planets in our cosmic neighborhood. Looking ahead, the upcoming Extremely Large Telescope is expected to enhance exoplanet research, potentially allowing for the detection of more small, rocky planets in temperate zones around nearby stars. Recent research has unveiled groundbreaking insights into gamma-ray emissions during tropical thunderstorms, including the identification of long-duration gamma-ray glows and a newly recognized phenomenon known as flickering gamma-ray flashes, or FGFs. This study was conducted by an international team from the University of Bergen in Norway, in collaboration with NASA and various universities. The research involved 10 flights over tropical storms using a modified NASA ER-2 aircraft, where scientists observed gamma radiation in 9 out of 10 storms. During these flights, approximately 500 glows and 130 terrestrial gamma-ray flashes were detected, suggesting that such emissions are more common than previously thought. The findings indicate that over half of all tropical thunderstorms emit radioactive gamma radiation, with these emissions being dynamic and varying in intensity, similar to steam escaping from a boiling pot. Published in 2 studies in Nature on October 2, 2024, these results have been described as game changers for the field by physicist Joseph Dwyer, who emphasizes the necessity for further research into these phenomena. This research challenges existing theories regarding lightning initiation, proposing that positron feedback may be responsible for the observed gamma-ray emissions and their connection to lightning, rather than cosmic rays. Notably, most lightning strikes were recorded shortly after the glows and flickering flashes, suggesting a potential link between these gamma-ray phenomena and the initiation of lightning. The glows were found to be unexpectedly persistent, covering thousands of square kilometers and lasting several minutes, while FGFs were shorter-lived, lasting between 50 to 200 milliseconds. Despite the presence of gamma radiation, the levels detected pose no immediate danger to people or aircraft, which typically avoid flying through active thunderstorm cores due to turbulence. The study was part of the Aloft campaign, aimed at observing terrestrial gamma-ray flashes associated with lightning, furthering the understanding of the relationship between gamma radiation, thunderclouds, and lightning. These findings could provide crucial insights into the long-standing mystery of lightning initiation, as gamma-ray radiation is linked to extreme cosmic environments. Age-related macular degeneration, or AMD, is a leading cause of irreversible vision loss in the United States, impacting up to 15 million Americans. Currently, there are no treatments available that can stop the progression of AMD, particularly affecting individuals over the age of 50. Existing therapies have limited effectiveness and can lead to significant side effects, making the need for new solutions critical. Researchers have identified a key protein involved in the early stages of AMD by examining genes related to the disease and macular dystrophies. The study, led by Dr. Ruchira Singh from the University of Rochester, utilized human stem cells to model AMD, addressing the limitations of previous animal model studies. This innovative approach has allowed researchers to explore the underlying mechanisms of AMD more effectively. Targeting inflammation pathways linked to a protein called TIMP3 has shown promise in reducing the formation of drusen, which are lipid protein-rich deposits that accumulate in the retinal pigment epithelium and serve as early indicators of AMD. The findings from this study could significantly enhance treatment strategies for millions affected by AMD, as halting drusen accumulation may prevent further vision loss. Published on October 2, 2024 in the journal Developmental Cell, this research offers hope for those impacted by AMD. The study was backed by the National Eye Institute and other organizations, highlighting a collaborative effort to find new therapeutic targets aimed at potentially halting the progression of AMD. A groundbreaking study published in Nature on October 2, 2024, sheds light on the decline of neural stem cell activity in aging brains. Conducted by researchers at Stanford Medicine, the study reveals that the knockout of glucose transporter genes, specifically GLUT4, in older mice, led to a significant increase in the production of new neurons. This finding uncovers a potential pathway for rejuvenating these crucial cells, which are vital for memory and overall cognitive function. Neural stem cells in the adult brain can generate new neurons. However, their effectiveness diminishes with age, contributing to cognitive decline and neurological disorders. Utilizing CRISPR technology, the researchers identified 300 genes that, when disrupted, could enhance neural stem cell activation in aged mice. This indicates a complex genetic landscape that influences neurogenesis, emphasizing the need for targeted research in this area. The study highlights the significance of understanding which genes can enhance neural stem cell activation, potentially leading to interventions that counteract age-related cognitive decline. In the subventricular zone, a critical niche for these stem cells, the transition between quiescent and activated states is impaired in aging brains, correlating with cognitive deficits. Furthermore, the findings suggest that interventions focusing on glucose metabolism, including dietary changes such as low-carbohydrate diets, may improve neurogenesis in both aging and younger brains. The research also uncovered pathways related to primary cilia that may influence neural stem cell activation, hinting at a connection between glucose metabolism and brain health. These insights are pivotal for developing cell therapies aimed at treating neurodegenerative diseases, as they open new avenues for promoting neuron growth in older or injured brains. The GLUT4 gene, crucial for glucose transport, has been identified as a key factor in keeping aged neural stem cells inactive, underscoring the role of metabolism in brain health. The study received funding from the National Institutes of Health and other organizations, supporting the exploration of genetic and dietary approaches to enhance neural stem cell activity. Future research will focus on the effects of glucose restriction in living animals, providing a deeper understanding of its impact on neural stem cells. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.